Thanks, Jeremy. Thanks for inviting me to do this talk. So I'm talking about an area of medical mycology which I'm really passionate about. I really love um, dimorphic fungi, not so much the fungal infections they cause, but really the pathogens are truly fascinating. Um, and so I'm going to be telling you about just two age-related dimorphic fungal infections. And I thought I'd start with my take-home messages. So if you want to, um, you know, drop off directly thereafter, you can. So really, histoplasma and emergomyces are the pathogens we're going to talk about this afternoon. They cause clinically indistinguishable syndromes in patients with AIDS, and they're treated in more or less the same way. The two fungal genera are very, very closely related from a genetic point of view, and they both contain cryptic species, some of which are unique to Africa. The yeast forms of histoplasma and emergomyces are almost completely indistinguishable, with one exception, which I'll tell you about later. But the mold forms can be distinguished in culture. There are rapid histoplasma antigen tests which cross-react with emergomyces, and these really should only be ordered when you're trying to diagnose symptomatic disease, disseminated disease in patients with advanced HIV disease. And then we've got some investigational PCR assays, which seem to be most useful on samples from the infection site. Okay, so let's take one step back and talk about fungal dimorphism. So fungal dimorphism is a really interesting topic. And actually, many fungi have the ability to transform from one form to another. And the triggers for that transformation really vary. So many of you will know the fungal genus Mucor, which causes mucormycosis. Interestingly, most of you will recognize this pathogen as a mold, but in the presence of decreased amounts of oxygen, the pathogen can exist as a yeast, and with increased oxygen concentrations can transform to a mold phase. Malassezia furfur, which causes tinea versicolor, or pityriasis versicolor, is also typically a mold, but also in the spaghetti and meatballs appearance that you see on skin scrapings, shown on the slide here, you get a morphological transition in the presence of L-dopa. And then tinea nigra, which causes, Hortia wernickeyi, which causes tinea nigra, which is these hyperpigmented papules, usually on the palms and sometimes the soles, transforms from a yeast to a mold in the presence of increased salt. But fungal thermal dimorphism is a much rarer phenomenon. Sorry. So fungal thermal dimorphism is a much rarer phenomenon. There's 7 fungal genera for which temperature is the trigger for conversion from a mold to a yeast form. All of these fungi belong to the Ascomycetes group. And these fungi are shown on the slide. So the fungi include coccidioides, paracoccidioides, Tallaromyces, histoplasma, emergomyces, blastomyces, and emergomyces, and Sporothrix. So those are the fungi number 1 through to 7. Anyone want to guess which was the first described thermally dimorphic fungus? If you put it in the chat, I'll have a quick look. Okay, so this is going back about 120 or 130 years ago. And in fact, blastomyces was the second thermally dimorphic fungus to be discovered. First was cocci, detected in 1892, and then about 6 years later, blastomyces, and then sporothrix and histoplasma only in 1905. And the fungi which we're talking about today, histoplasma and then later Emonsia, which is now renamed emergemyces, detected in 1960 and 1992. I thought I would use one of Jeremy's cases as a sort of a framework for this discussion. So, a 41-year-old man with advanced HIV disease on fixed dose combination ART for a week before he presented to hospital. His baseline CD4 count was 63, and he had a very high viral load. He had a 3-week history of skin lesions on his face, neck, and forearms, and these were associated with sores in his mouth and on his lip. Cutaneous lesions were not pruritic, not tender, but the oral lesions were very, very, very painful, and he had difficulty eating owing to the pain. He'd had previous episodes of similar lesions over the previous 7 months, but these had resolved without treatment. He previously had TB meningitis in 2013, for which he'd received 9 months of treatment, and he'd had a previous appendectomy. He was originally from Ethiopia and had moved to South Africa 12 years before his presentation and hadn't gone back to Ethiopia since then. He lived in Mayfair in Johannesburg and worked at a shop in the city, and he had no other relevant habits. No pets, no animal contacts. And really on examination, the major features were that he was wasted, and he had these ulcerating lesions on his lips, tongue, palate, as well as sporal thrush, and multiple erythematous papules and nodules on his face, neck, and forearms. So these are the pictures of those lesions. And you can see the suggestion of this one in particular being umbilicated, possibly. Here's a close-up, or maybe that was just, maybe he'd scratched it or something. And here's a picture of the oral or the lesion on his palate, which was very painful. You can also see that he had oral thrush. So on examination, further examination, nothing else really to find apart from frothy lymphadenopathy. So the differential diagnosis from an infectious point of view includes, and through these regular meetings, I know we've gone through this differential diagnosis before, includes bacterial causes such as secondary syphilis, TB, and other mycobacterial infections, bacillary angiomatosis, from a fungal point of view, crypto, emergo, histo, from a parasitic point of view, cutaneous leishmaniasis, and from a viral point of view, molluscum contagiosum, particularly with umbilicated lesions. So, what do we know about emergemyces and histoplasma? Well, emergemyces and histoplasma causing these two diseases. I'm going to take you just a step back, tell you a bit about a condition called mycologist, which is a really unusual, very uncommon disease. It's caused by a pathogen called Emonsia crescens, which is a soil saprophyte. And it was first described in animals, in rodents in Arizona in 1942, described by Emmons. And the pathogen is named after Emmons. The genus is named after Emmons, Emonsia. The first human case occurred in France in the 1960s. And essentially, what happens is you inhale these canidia. The term Aedes spiromycosis comes from the adiocanidia of this fungus, which essentially progressively enlarges in the lungs without replication. at 37 degrees Celsius. So the inhaled conidium, which starts off at about 2 to 4 microns in diameter, can actually grow to 200 to 400 microns or more, which is a million-fold expansion in its volume. And patients develop pulmonary lesions, which are produced by these enlarging conidia. They tend to be these discrete or confluent granulomas, each containing these thick-walled, spherical adiaconidia. And in fact, they're just large enough to be able to be seen with the unaided eye. So the granulomas maintain a distribution in the lungs around the bronchioles, which indicates, of course, that there's an inhalational portal of entry. And the symptomatic disease is really due to the inflammation and fibrosis, which happens because of this granulomatous inflammatory reaction. So this is ADS spiromycosis, really rare fungal infection, pulmonary fungal infection. And I told you that Emansia crescens was detected first in 1960. And for the next 30 years or so, there were no reports of any disseminated disease cases. In 2013, we then described a series of cases of disseminated emansiosis, which really shifted the paradigm for this disease entity, because it was clear now that Emansia-like fungi could cause not just a localized pulmonary disease, but also disseminated disease, and not just with these Aedes spores, which didn't replicate in vivo, but now also with small budding yeast cells, very much like histoplasma, which were found in vivo and weren't restricted just to the lungs, but disseminated much more widely. But really, this changed the paradigm. And it led to a taxonomic reshuffling within this entire group of fungi called the Agelomycetacea. So a new genus was set up or created to accommodate this new group of dimorphic fungi, causing disease predominantly amongst immunosuppressed human patients. It was so named because of the apparent emergence of these closely related pathogens around the world, and not, and of course it's an apparent emergence because, you know, this fungus has been around for probably hundreds, if not thousands of years, and the emergence is more a consequence of the refined molecular diagnostic techniques that we'll be using to diagnose patients, rather than a truly recent emergence. So within the Emergomyces genus, there was an initial separation of Emansia crescens from the other Emergomyces species. So there were originally 5 Emergomyces species: Canadensis, Orientalis, Pastorianus, Europaeus, and Africanus. And then Emansia crescens was kept separate. And if you look at the morphology of these fungi, you'll see that Emansia crescens has those very large Aedes spores at 37 degrees Celsius, shown in this picture on this phylogenetic tree. And as you go down towards the Emergomyces species, you see more the typical budding yeast cells. But ultimately, on further taxonomic examination, and the fact that there probably is quite a wide morphological variation within this genus, the entire Emansia genus was collapsed, and Emansia was then reclassified as Emergomyces. So 2 remaining species were then moved to Emergomyces, and Emergomyces now has 7 species in total. So unfortunately, that Emansia genus that was named after Chester Emmons, a really eminent US mycologist, is now no longer taxonomically valid. So emergomycosis is a globally distributed fungal disease. There are cases on 4 continents: Europe, Asia, Africa, and the Americas. And So it includes, as I've said, Emergomyces posteriornis in Europe, Asia, and Africa, Canadensis in Canada and the US, Europaeus in mostly Germany so far, Orientalis from China, and Africanus almost exclusively as far, no, Africanus from South Africa, Lesotho, and Zimbabwe. So Southern Africa's got 4 endemic mycoses that we know of to date: Sporotrichosis, histoplasmosis, blastomycosis, and emergomycosis. And we've analyzed pathology lab data from public sector labs and some private labs in South Africa over roughly a decade. And when we looked at the laboratory databases, we found about 680 odd cases, 45% of which were laboratory confirmed cases of disease where the pathogen could be assigned to one of these 4 genera. And then there were about 41% which were probable, 14% possible cases in that series. And this just shows you the number of cases which were diagnosed per year between 2010 and 2020. So this was part of the PhD work conducted by Rutendo Mapengo, who's been working with us on histoplasmosis. And you can see that there was a bit of a spike probably around 2014, 2015, 2016. That was around the time that we published that case series of what we call disseminated monziosis. And so there was a lot of interest in case finding, I guess, around emergomycosis. So many more cases, I think, were diagnosed. The most common endemic mycosis we think in Southern Africa is sporotrichosis, not any of the others. So what is the presumed pathogenesis of emergomycosis? We think it's similar to histoplasmosis, but we don't know for sure. And of course, similar to blastomycosis too. So these tiny microconidia, which are found in the mold phase, are inhaled. They bypass the structural defenses of the lung, such as the ciliated epithelium. They enter into the lower respiratory tract and lodge in the terminal alveoli. And they convert to the yeast form, which is really triggered by the body temperature. And that's a histidine kinase mechanism. They then bind to the innate immune cells, phagocytosed, they germinate and they replicate inside cells as budding yeasts, and they're quite happy inside the cells. And they then disseminate predominantly via the bloodstream, but also the lymphatics. So, based on this hypothesis, where is Emergomyces africanus found in the environment? So we searched for the reservoir, and this was a PhD work that was conducted by Ilan Schwartz when he was based in South Africa about 10 years ago now. And so we were searching for the reservoir based on the assumption that the reservoir was the soil. And Ilan collected a convenient sample of soil, soil samples from around the country. Most came from the Western Cape because that's where he was based. And these soil samples were examined using a nested PCR assay using ITS primers, and then followed by some more specific primers amplifying a smaller region just to be sure this was Emergomyces. And the PCR was pretty sensitive. It could detect as few as 100 conidia in 10 grams of soil. So these were some of the soil samples. We tried to culture the fungus from the soil, but it really didn't work because of the rapid overgrowth by contaminants. We used the flotation method to create a soil suspension that separated the conidia from the substrate and then hopefully the contaminants. But when the suspension was plated, it still demonstrated the presence of the contaminants, so that didn't really help. We also tried passaging the soil suspension through mice, and then we cultured their spleens and livers after 2 weeks. But that didn't work either, unfortunately. But we did find evidence of emergomyces in the soil from a molecular point of view. 30% of samples were positive for emergomyces africanus by PCR. And in this figure, the black dots indicate where emergomyces africanus was detected in the soil. The white dots is where it was not detected. And then the crosses represent the residential postcodes of the cases. Ilan did all this mapping work to try to work out if there was any correlation in sort of space and time, but nothing clear there. We also couldn't find an animal reservoir. You know, for many of the endemic mycoses, there's also an animal reservoir, I guess most famously for talaromycosis, where the animal is gone completely blank. It'll come back to me. So there's a well-known animal reservoir for talaromycosis. So completely forgot. So we tested the lungs of 1,400 animals, representing 26 species collected across South Africa. This included 4 species of mole rats because we thought those would be the animals most likely to be infected because they burrow into the soil. And so we used PCR methods again, but unfortunately, none were positive. Ilan also did some additional work looking at emergomyces spores in the air, and he set up these spore traps in Cape Town and analyzed the spore strips for DNA of emergomyces africanus using a PCR method. And interestingly enough, emergomyces africanus was detected on 10% of the days during which sampling was conducted. And these tended to be cooler days, days where Cape Town was less windy, and also days where Cape Town was rainy. So cooler, rainy days with less wind. So the known spectrum of clinical emergomycosis is limited, I would say. So far, we've got no known cases in immunocompetent hosts. All the original cases we thought belonged to disseminated or were cases of disseminated emonsiosis in immunocompetent people turned out actually to be cases of blastomycosis. Emergomyces africanus has most commonly been reported to date in Southern Africa, almost exclusively among patients with advanced HIV disease, and causes a multi-system disease. Emergomyces posteriornis, canadensis, and orientalis affect people with a variety of background immunosuppressive conditions, and they tend to cause multi-system disease also. There are a couple of cases of isolated pulmonary involvement. So from Germany, Emergomyces europaeus has caused there's a patient with limited pulmonary disease. And then in Canada, a patient with Emergomyces posteriornis. Now, CNS involvement seems to be incredibly uncommon. And so to date, we've not found any cases of meningitis or space-occupying lesions. Not to say that they don't exist, but they've just not been described. And so just to make the point that the clinical spectrum of emergomycosis really reflects, I think, the tip of the iceberg, or from an African point of view, the proverbial ears of the hippo. So what you see, what is visible above the water is really disseminated disease in AIDS. And of course, that's a very dramatic presentation. So particularly with the skin lesions, people know to recognize that syndrome, and so cases are diagnosed. But we think that there are probably other as yet unrecognized clinical syndromes. I mean, probably many more cases of isolated pulmonary involvement, for instance, which are just never detected because they might be misdiagnosed as TB or another disease. So in terms of the the clinical manifestations of emergomycosis. In the various case series that we've looked at or published on, the median CD4 count was usually around 16. Cutaneous lesions were by far the most common clinical feature, present in about 96% of patients. But that, of course, is a diagnostic bias. And they frequently appeared as these plaques or papules or ulcers. And pulmonary lesions were also fairly common, 87% of cases with radiological involvement. Just to say also that emergomycosis and comorbid TB is fairly common in Southern Africa, as you'd expect. I also didn't want to forget about histoplasma. So histoplasma capsulatum is, well, I mean, it's called histoplasma capsulatum, but contains multiple cryptic species, is also found in the soil. I told you about the pathogenesis because it's exactly the same, we think, for emergomycosis. But the ecological reservoir is a bit better known for histoplasma because of a lot of work that's been done predominantly in the Americas, in the hyper-endemic zones in North America in particular, where histoplasma has been found in soil associated particularly with bird and bat guano. But histoplasma actually is a cosmopolitan fungus. It's found across the world, and you can see that this new map, which I've sort of pasted onto the CDC life cycle diagram, shows that it's actually not restricted to the Americas, but actually is found in most of Africa and probably in Southeast Asia too. And there certainly are cases which occur in some parts of Europe, predominantly Southern Europe. So let's go back to our case. I took a long break there, but I'm just now showing you some of the laboratory investigations from this patient. He had most of these results are normal, just the ones indicated in red, abnormal, so very high serum ferritin, probably indicating chronic inflammation. He had a low albumin, elevated ALP, GGT. Just to note that his blood cryptoantigen test was negative. And that's really important because, of course, crypto is on the differential diagnosis, as we mentioned earlier on. The patient also had a chest X-ray, which was normal, and an ultrasound, which was a bit vague. Well, the report was a bit vague. Okay, he did have a skin biopsy. And of course, that's a really useful investigation. And ideally, the skin tissue should be sent for both histology and for culture. It was sent for culture, for fungal culture, but there was no growth. TB culture was not done, unfortunately, because the specimen leaked, and so it was unsuitable for testing. But in histology, they noted tiny spherical organisms within some of the macrophages, which they thought was suggestive of the amastogotes of leishmania. So the patient was started on amphotericin B deoxycholate, which is appropriate treatment for leishmaniasis, with significant clinical improvement. But of course, this is I'm telling you about histoplasmosis and emergomycosis. So, this is one of those two diseases. And so just talking about how urgent is this diagnosis compared to other fungal clinical syndromes. So usually disseminated histoplasmosis and emergomycosis are subacute or chronic presentation. Of course, with cryptomeningitis, rhinocerebral mucormycosis, candida sepsis, you need a rapid diagnosis, ideally within hours to days. For disseminated histoplasmosis, emergomycosis, you've got a bit more time. Obviously not weeks and weeks because the patient will deteriorate. But you probably have, you can try to confirm the diagnosis, before starting antifungal treatment. You might have some time if the patient is clinically stable. Just to say that in a small cohort study, a small cross-sectional study, actually, it wasn't a cohort, of 37 patients, where the case fatality ratio was about 48%, we found that lack of skin biopsy samples was actually associated with death in patients with emergomycosis. And this, of course, is a possible proxy for delayed diagnosis, because if someone does not have the skin lesions, those typical skin lesions, then clinicians are less likely to think about emergomycosis or histoplasmosis. So if you've got emergomycosis diagnosed only, for instance, on positive blood culture or positive bone marrow culture, the outcomes might be somewhat worse. So the histology report was reviewed, and the Pass and Grocott stains highlighted small yeasts, both intracellularly and extracellularly, with apparent narrow-based budding. There were no hyphae observed. So this was clearly a fungal cause. But remember, the initial diagnosis was leishmaniasis. So I think they sent this through to Monica Burkhead, who's actually on the call. I think I saw her, spotted her name somewhere on the participant list. And Monica did, created these beautiful pictures to definitively exclude leishmaniasis co-infection. And so this here is of the pathogen, just various views of it. And so this was Jeremy's saying this is clearly not a virus. It's far too large. There's a nucleus, so clearly not a bacterium. Prokaryotes don't have nuclei. It's got a cell wall, so it's not a protozoa or helminth. And it's clearly, remaining life form. It belongs to the kingdom fungi. So, just to go to my back to my take-home message, histoplasma and emergomyces yeasts are indistinguishable on histopathology. I've shown you there are 3 different stains on this slide. There's the classic H&E stain, which is just blue and pink. The bottom left is the stain, the periodic acid Schiff stain, and here the budding yeast appear as this bright magenta pink. It stains the carbohydrate in the cell wall of these fungi, and then you've got the Grocott or the methenamine silver stain at the bottom right, and here the fungal elements are stained up as the blackish brown color. So remember, this patient was from Ethiopia. He'd relocated 12 years ago, and he moved to South Africa and hadn't returned. So important, of course, to think of what is endemic in Ethiopia, what's endemic in South Africa. So I've just listed some of the fungal diseases which are reported from each of these countries. I just wanted to mention a little bit about some of the test level barriers for emergomycosis and histoplasmosis. So in many places in the world, particularly in resource-limited settings, the gold standard diagnostics are not widely available. And the gold standard diagnostics, of course, are microscopy and culture from blood, bone marrow, and skin tissue, etc., and also then the histological examination of that tissue. And so rapid antigen testing on urine and serum is much more feasible. At the moment, we predominantly have these antigen tests for, in an enzyme immunoassay format, and that requires laboratory infrastructure. There are now some lateral flow assays, either in development or available but not on the market. So, and there's one other lateral flow assay which doesn't perform particularly well for histoplasma. I'll come back to discussing the antigen tests, just to say that that histoplasma and emergomyces yeasts are also indistinguishable on Giemsa-stained peripheral blood or bone marrow aspirates. This was a slide from a patient with emergomyces infection, not from this particular case patient. And you can, this looks exactly like you would expect for a patient with histoplasmosis, disseminated histoplasmosis. And then, of course, if you look at a gram-stain smear of a positive blood culture, the yeasts, again, are going to be indistinguishable on gram-stain smear. There's one exception. So this is the pathogen called Histoplasma capsulatum var. duboisii. And this is African histoplasmosis, or the cause of African histoplasmosis. Now, this was described, if I remember correctly, in the 1930s or 40s. No, I'm sorry, I think maybe in the 1950s. These were large budding yeasts of 7 to 15 microns in diameter, so very different from the classic emergomyces and histoplasma yeasts, which are much, much smaller. They're only 2 to 5 microns in diameter. These are much bigger. They look like blastomyces, actually. They're ovoid, and they also have very thick cell walls. And what you also see often is formation of chains, sometimes in the shape of an hourglass, or sometimes a figure 8, and you can see lots of figure 8s in this picture here. And sometimes also you see these cytoplasmic lipid inclusions, which most of the cells in this picture also have. So it's quite easy to recognize Histoplasma duboisii in fluid specimens, but not so easy to recognize the morphology in tissue. So I used to think, and I think this was sort of the classical teaching, that in Southern Africa, we didn't see Histoplasma duboisii. It was mostly capsulatum. But these authors from KZN published, histopathologists from KZN, published a couple of years ago in the Journal of Cutaneous Pathology, a series of 24 cases of what they call Histoplasma capsulatum var. duboisii. And I initially thought this was unlikely to be duboisii, but on looking at the paper in a bit more detail, so these were all patients with advanced HIV disease. The median CD4 count was 38. 70% of these patients were on ART, all had skin manifestations, and 8 of the 24 actually had culture positive confirmation of histoplasma infection. So what you see on the skin biopsy, and you can see it's a Grocott stain, is a mixture of small and large yeasts. They're different shapes, and they also in the publication they also draw some of the shapes that they saw. They also did PCR directly from the formalin fixed paraffin embedded tissue, and they, based on that, it was pretty much an ITS PCR. Based on that, decided it was Histoplasma capsulatum var. duboisii, which I think is really interesting. Let's go back to the patient, our case patient. So this patient from Ethiopia, with those skin lesions and the oral painful lesions, had a histoplasma antigen test which was negative. All his other TB workup were negative. I mentioned earlier that antigen tests are better than the gold standard in terms of access, but you can see when you compare to the cryptoantigen test, so the map on the right shows you access to cryptoantigen tests, map on the left, histoplasma antigen, and you can see really just from a color coding point of view that the crypto test is much more widespread, much more easily available. The histoplasma antigen assay needs a lot needs to be done before this assay will become available. It is on the list, the model list of essential in vitro diagnostics. WHO included this on the EDL about, I think it was 3 or 4 years ago. No, in 2020, actually. 4 years ago now. So just to say that we've done some work in terms of evaluation of this a particular commercial histoplasma antigen test in South Africa. And of course, our setting is co-endemic for histoplasmosis and emergomycosis. And so what you can see is that when you use culture-confirmed disease as your reference, culture-confirmed histoplasmosis as the reference, the test sensitivity is 88% and the specificity is 72%. The specificity is lower because, of course, there's some cross-reaction with emergomycosis or emergomyces. When you include both emergomyces and histoplasma infection, you can see the sensitivity drops because this test really targets histoplasma rather than emergomyces. But then the specificity increases to 91% because you don't really care whether it's emergomycosis or histoplasmosis. From a clinical point of view, they're pretty much the same disease as far as we can tell. We've also done some work together with Institute Pasteur, developing some real-time PCR assays. So we've developed 2 additional assays. The Institute Pasteur group developed a histoplasma-specific PCR, and you can see that when you test, and then we also then developed an emergomyces-specific PCR, and then one that picks up both the emergomyces and histoplasma, which we call it our pan-dimorphic PCR because it also cross-reacts with blastomyces. So you can see that when you test these 3 PCR assays against histoplasma capsulatum, CT values are lowest for histoplasma because it's designed for histoplasma, and then you get higher CT values for when you apply the emergomyces PCR and mixed PCR assays to histoplasma capsulatum. And then similarly, if you apply the emergomyces specific PCR assay to emergomyces, you see the lowest CT values for emergomyces, and then when you apply the histoplasma and mixed PCR assays to emergomyces, the CT value is higher. And then, of course, as I said, all these assays tend to cross-react with blastomyces. Just in terms of a diagnostic point of view, we think that these real-time qPCR assays may actually work best on samples close to the site of infection. The reason we say this is because, we looked at about 27 cases, culture-confirmed cases of emergomycosis and histoplasmosis. And when we stored urine samples from these patients, and, most of these patients had skin lesions. The sensitivity in urine was only 26%. We didn't have skin tissue in order to then, perform the RT-qPCR assay. But when we did look at histology-confirmed cases where we did have the tissue, the sensitivity was 100%. So if you actually send us a sample from the site of infection, we think this PCR assay would work really well. It doesn't seem to work quite as well on urine. Okay, so back to the case patient. A fungus finally was grown. So this was a second biopsy because remember the first one, nothing grew on the first. And so what you see here is a fairly slow-growing fungus, which is sort of cream-colored. It's a mold, cream-colored to brown, surface and reverse. It grew extremely slowly. And when the microscopic appearance was looked at with using a lactophenol cotton blue slide preparation, it was confirmed to be histoplasma capsulatum, and it was confirmed to be capsulatum based on ITS sequencing. Now, those of you who are familiar with fungal morphology will see this is an atypical histoplasma slide, because in fact, what you normally see are these macroconidia which tuberculate. And you can see here, these are smooth. They don't have the spiky projection that you expect to see. I'm not sure why this particular fungus had lost its tuberculate projections, but that is in fact the case. And then just to say some additional work from our team. So Histoplasma capsulatum, we know, contains cryptic species. So there've been some studies previously completed, published back in 2017, where whole genome sequencing was done of a fairly large collection of histoplasma strains, or looking at the whole genomes. And this is a phylogenetic tree, and you can see that within this tree there are monophyletic clades, which are probable cryptic species. So they've all been given names now. And in fact, the only one that didn't get a name is the Africa cryptic species, because in fact in this study there were only 2 African isolates, and so there really wasn't sufficient data to say that the African clade was separate. But now Ritenda's done some new work as part of her PhD, which she'll hopefully be submitting in the next week or so. Here she included 36 isolates from all over the African continent. And you can see here that very clearly the African clade is separate and is most closely related to Panama, the Panama strain. So interestingly enough, this clade incorporates both var capsulatum and var duboisii. So even though the yeasts look morphologically different in body fluid samples, when you look at the 2 varieties, capsulatum and duboisii, from a genetic point of view, they are actually indistinguishable within that phylogenetic step. Emergomyces can be distinguished in culture. So this is just a picture of Emergomyces africanus, the mold phase. This is the yeast phase. Then 4 to 6 weeks. And we've also, we've got another scientist in our team who's been looking at whether there are cryptic species within Emergomyces africanus. When we initially published this paper in the NEJM back in 2013, we generated a phylogenetic tree using 5 genes. And so it was, these fungi looked precisely, well, they looked almost exactly the same. So we thought we'd use whole genome sequencing to see if we could get better resolution. And Slindiwe has been doing this work over the last 2 or 3 years, painstakingly culturing roughly 93 or so isolates. She'd looked at the morphology of all these Emergomyces africanus isolates, and she's found 2 different phenotypes from a mold point of view. 90% of the isolates form these wrinkled or cerebriform colonies, which are pale and become slightly darker, whereas 10% of the isolates are flatter and less wrinkled. From a yeast point of view, for the yeast phase, there's no real difference. She also looked at the whole genome phylogeny, and I'm not going to take you through the details, but just to say that there are definitely groups within Emergomyces africanus, 8 major groups, in fact. And interestingly enough, the first cases we reported back in 2013 all cluster within group 1, and thereafter there's been some shift in their genome composition, I suppose.
I'm sorry about that. We seem to have lost Nelesh. 1 second. Let me just see if I can get him back. All right, Nelesh is just logging in. Let's get us back. Sorry about this. Seem to have lost internet at our venue. 1 second, we're back.
Okay, and I'm back. Sorry about that, everyone. I'm at Helen Joseph, and Jeremy, yeah, or our internet connection just dropped completely. Sorry, I'm going to just share my screen again. Must be honest, I'm not certain where I got cut off, but I'll just say that Cindy Way essentially found potent activity against amphotericin B, the mold active azoles, but not fluconazole and certainly not echinocandins and flucytosine. So in terms of this patient's management, he was discharged on high-dose fluconazole because I don't think itraconazole was available at the time after completing 20 days of deoxycholate amphotericin B. This was gradually reduced to 400 milligrams of fluconazole, although he initially worsened and only responded when the fluconazole increased again to 800, which suggests, I think, that this histoplasma isolate probably had relatively high fluconazole MICs, but sufficiently low for fluconazole to work. His CD4 count improved on ART, and he suppressed his viral load within 4 months or so. And you can see there was skin lesions on admission, and these were his skin lesions or his skin on discharge and at the ID outpatient clinic 2 weeks later. Really nice to see such a good outcome in this particular patient. And then I'm just going to end by saying a few things about what do we know about the natural history of histoplasmosis and advanced HIV disease. Well, the bottom line is we don't really fully understand it. We assume there's reactivation, but certainly some new infection. We don't know how long the period is between reactivation or infection to symptoms, how long from mild illness to severe illness, and we don't really know when we can start detecting antigen or even nucleic acids in body fluids. So, given that we don't know the natural history, it's quite difficult, of course, to consider something like screening. So, this slide just shows you published reports of histoplasmosis in Africa. The vast majority of cases diagnosed in Southern Africa, and also you can see in Nigeria, more than 100 cases, total more than 400 cases reported in the literature over 7 decades. So, of course, this is a minimum estimate. So Rita Oladele and various others in Nigeria decided to look at whether one could use the antigen test to diagnose people with advanced HIV disease and a clinically compatible syndrome, of histoplasmosis. And they actually found that in this scenario, 8% of 988 patients had a urine antigen test which was positive. But just a warning that given that the positive predictive value is only about 67% of the true prevalence is 5%, you ideally should really be doing confirmatory testing before initiating antifungal treatment. I won't go through the details here. We have recently collaborated with Charlotte Schutz, Graham Mankies, and others from UCT. We looked at a cohort that Graham and Charlotte had set up, patients with advanced HIV disease and suspected TB, in whom all the TB tests were negative. And in fact, we found that none of the 89 urine histoplasma antigen tests were positive in this cohort. These are brand new data. The laboratory team has just finished all this testing. But of course, the prevalence might just be low, might just be below the threshold of our, the sample size that we have. So when should you really use the histoplasma antigen test in clinical practice? Don't use it when someone doesn't have symptoms. So you should not be using it like you do for cryptoantigen screening, because we don't know the natural history, as I've said, but it does seem reasonable to use it for diagnosis. There's lots of experience in the Americas, and particularly in Latin America. A key unanswered question we think is how to confirm the diagnosis, given that low positive predictive value in low prevalence settings. It seems possible that EIA units might be useful, but we still need to do a bit more work on that. And of course, we must use the antigen test for surveillance to understand the disease burden in HIV. And it's really exciting that Unitaid has just made some money available to various groups around the world, including Aurum in South Africa, to do some surveillance work to look at antigen testing to understand the disease burden in AHD. And through our imprint group, we're also doing similar work. So just to remind you, take-home messages again. Histoplasma and emergomyces cause this clinically indistinguishable syndrome. You treat them in much the same way with amphotericin B and itraconazole thereafter. The two genera are phylogenetically closely related. They both contain cryptic species. Some of them are unique to Africa, such as Emergomyces africanus and this histoplasma, I don't know, it doesn't have a name yet. And then the yeast forms of histoplasma and Emergomyces can't be told apart, but you can distinguish the mold forms in culture. And then the rapid tests, the antigen tests, cross-react with Emergomyces and blastomyces, and you should really only be using them to diagnose symptomatic patients with advanced HIV disease. And don't forget about those investigational PCR assays, but they seem to work better on samples from the infection site. So I'm just going to end there, acknowledging some of our team in the laboratory, but also our imprint group. Thanks.
Thanks so much, Nelesh. Really wonderful talk, and nice to get an update on these really impressive and interesting fungi. I just looking through the chat, I see Narcisa from the pediatric side is asking about the youngest age for emergomycosis or histoplasmosis in AHD. In other words, are we missing it in pediatrics, do you think?
Yes, I think you're missing it, Narcisa. There was one case reported from Cape Town in an adolescent, if I remember correctly. I think around 12 or 13. But there might well be other cases. So if you see a compatible syndrome, definitely think about histoplasmosis or emerging cases.